Ethylene, an important plant growth regulator that is associated with the ripening of fruits. Apart from it, which has many other functions in the plant body. So the today's topic for discussion will be ethylene phytohormone, where we'll see the discovery, its biosynthetic mechanism, signaling cascade, as well as the functional profile of this plant growth regulator. So let's proceed on to the video and understand this phytohormone in detail. So firstly, coming to the introduction of this phytohormone, so this ethylene is a hydrocarbon compound which has a molecular formula of C2H4, which is represented as two CH2 molecules that are linked together by a double bond. So you can see here it is linked by a double bond. It is a colorless gas which has a sweet odor, and that's why it is called as a gaseous hormone, which forces the ripening of fruits in plants, whereas the ethylene levels are more. So if you compare two fruits, the one that is ripened and the other fruit that is a raw fruit. So the ripened fruit will have more amount of ethylene, which forces the ripening of fruits in plants. And when this ethylene is hydrated, it is converted to ethanol. So the ethynyl hydrate is called as ethanol. So for the very first time, it was found in 1910 in ripe oranges. And the technique that was used for the identification of this phytohormone was chromatography. So who was the discoverer? It was Argane who discovered this volatile substance in 1934. So make sure this Argane, the scientist mentioned this ethylene is only a volatile substance. But this was identified as a phytohormone in 1935 by Cracker. So Cracker only called this ethylene as a phytohormone in 1935. So now let's see the biosynthetic mechanism of this phytohormone. So what's the precursor for the synthesis of ethylene? It's a methionine amino acid, which is a sulfur containing amino acid. But how methionine is formed in the plant cell? It is derived from cysteine amino acid. So this production of ethylene, it is present in all the organs of the plant. Means ethylene, it is present in all the plant cells as well as tissue. But if you want to see the maximum production of this, it's going to be the ripening fruits as well as senescent leaf. So shall we say this ethylene phytohormone is responsible for the ripening of fruits as well as the senescence of the leaves? Yes. So what's the precursor molecule? It's going to be methionine that is derived from cysteine. What is the location of this biosynthetic mechanism? It is taking place in plastid. So now let's see the biosynthesis mechanism of ethylene. So the biosynthetic pathway starts with the methionine amino acid, which in the presence of an enzyme that is adomate synthetase requires one molecule of ATP. You all know that synthetase are enzymes that occurs in the synthesis of some molecule utilizing ATP. That's why they are called as synthetases. Now you can see it's adomate synthetase, means adomate is going to be synthesized in the next reaction. So this methionine undergo a phosphorylation reaction in the presence of adomate synthetase, where it will synthesize S-adometh, or you can simply call it as SAM, which means 5-adenosyl methionine. This adometh is converted into 1-amino cyclopropane 1-carboxylic acid, which is also called as ACC. Now you can see here ACC is synthesized. That means the enzyme is going to be ACC synthase. So the second reaction is the synthetic reaction, where ACC is synthesized in the presence of ACC synthase enzyme. Now this ACC that is synthesized will undergo oxidation reaction in the presence of ACC oxidase that will lead to the production of ethylene. Now here half molecule of oxygen is utilized, which will lead to the ethylene biosynthesis, and along with it some byproducts are formed that includes carbon dioxide, hydrogen cyanide as well as water. So what happened in the biosynthetic pathway? Methionine is used as a precursor and it's converted finally into ethylene. So the very first reaction is the synthesis of the adomate, which uses an enzyme called as adomate synthetases. requiring one molecule of ATP. Once this adometh is formed, it is converted to ACC in the presence of the enzyme ACC synthase. And the last reaction being the oxidation reaction, where the ACC formed is oxidized in the presence of ACC oxidase, synthesizing ethylene along with some byproducts, that is carbon dioxide, hydrogen cyanide, as well as water. And now if you want to target this pathway, this ethylene biosynthesis, or if you want to stop the ripening of fruits at any stage, so you can just target the last two enzymes, that is ACC synthase as well as the ACC oxidase. Where you are targeting ACC synthase means you are stopping the production or the synthesis of intermediate, that is ACC. And if you are targeting ACC oxidase, that means you are halting the last step of the signaling pathway, that is the ethynyl biosynthesis. Now here we'll see the inhibitors that are involved in this pathway or that are used to target this signaling cascade. So the first inhibitor is AOA and the other is AVJ. So this AOA stands for amino oxyacetic acid and AVG stands for amino ethoxyvinyl glycine. So both of these inhibitors are used to target the second step of the signaling cascade, or you can say the second enzyme, that is ACC synthase. Now if this enzyme is inhibited, will there be a production of ACC in the plants? No. And if ACC is not produced, that means no production of ethylene. So it inhibits the ethylene synthesis. The second inhibitor is cobalt, that is CO2+. So it is used to target the enzyme, that is ACC oxidase, which is the ultimate step of ethylene biosynthesis. And if this enzyme is inhibited by this inhibitor, it will inhibit the ethylene synthesis. Now there are some stimulants which are used to stimulate both the enzymes, that is ACC synthase as well as ACC oxidase. So if stimulants are used for both the enzyme, what would be the result or inference in the plants? So if ACC synthase is stimulated, it will promote the ethylene biosynthesis. It also helps in the wounding and chilling process in plants. It enhances fruit ripening process. It also regulates the flower senescence, where it will promote the senescence or aging process in plant. It is also responsible for regulating the abiotic stresses in plants like drought stress as well as flooding stress. But if ACC oxidase is stimulated, so it will cause the ethylene production to be increased in plants. So it promotes ethylene synthesis as well as synthesis inside the plant cell. So this is all about the biosynthetic mechanism, where we have seen the pathway, the precursor involved, the location of the pathway, that is plastid, as well as the inhibitors and the stimulants for both the enzymes that are important for the ethylene biosynthesis mechanism. Now let's see the signaling pathway as well as the function of this phytohormone. Now let's firstly see the functions of ethylene biosynthesis. So once the ethylene is synthesized, it will serve many functions in the plant. So the first function is epinesty. What is epinesty? Epinesty is the downward movement of the leaves in the presence of ethylene. So once the ethylene is synthesized, it will cause the leaves to bend downward, and this is seen in case of tomato plant. And the second is the triple response in etiolation. This is one of the very important function of ethylene, for which ethylene is known. Triple response. How? It was first time recognized in the plant, that is Arabidopsis. Now what happens in triple response? So you all know that the seed which undergoes germination, and from this the very first leaf that is produced in the plant is cotyledon. It is called as cotyledon. Now this leaf will be present in the plant. Suppose these both are cotyledon. So above the cotyledon is called as area, which helps in the elongation of stem. So this area is called as epicotyle, and below this cotyledon, the area is called as hypocotyle. So this triple response is seen in case of hypocotyle. So what will happen? Naturally, the epicotyle and hypocotyle length will be normal. But if there is a high production of ethylene in plants, so this will act on this area, that is hypocotyle. And this hypocotyle and epicotyle, that is above and below the cotyledon, it serves for the elongation of stem. So what happens if any of these area, that is epi or hypocotyle, is inhibited? So what will happen? It will result in the reduced elongation of a stem and the stem height will be small. So if there is a large amount of ethylene in the plant cells, this will result into the reduced hypocotyle. That means inhibition of shoot or inhibition of stem elongation. So they will target this hypocotyle and the stem elongation will be reduced. Means the stem height will be less. And it also results in the increase in the width and thickness of the stem. The stem will be reduced but it will be thickened. That means swelling of hypocotyle. So we have seen that hypocotyle will be short and it will be thickened. Means chota hoga or mota hoga. Kaun? Hypocotyle. And the third response is tightening of apical hook. So what happens when seed is germinated? There happens a tightening. You have seen this curling of leaves. So this is what called as apical hook. So apical hook is a developmental program that happens in the process of germination in plants. So this apical hook that is formed in the presence of ethylene, it gets curled. That is what called as tightening of apical hook. So in the presence of ethylene, three responses are seen. That is what called as triple response. Firstly, the hypocotyle height will be reduced, which will result into the reduced elongation of the stem, and the stem will be thickened. That means mota hoga. And the last is the tightening of the apical hook. That is curling of apical hook. Now the next function is the fruit ripening. It also helps in the fruit ripening process as it's a gaseous hormone. It promotes the senescence in leaves. It also regulates the abscission process. What's abscission? It's the natural detachment of ripened fruits and leaves from the plants. So it regulates the phenomena of abscission, induces the lateral cell expansion, but it reduces the cell elongation rate. So these are all functions of ethylene hormone. Now let's see the signaling process. Now let's see the signaling mechanism. So the ligand that is used for the mechanism is ethylene itself. What is the name of the receptor? It's ETR1, which is also known as ethylene response 1, which is a copper ion based receptor that requires the need of the copper ion for its functioning. What is the location of the receptor? This receptor is located in the endoplasmic reticular lumen. What are the regulators involved? They are ETR1, ERF1, CTR1, EIN2 as well as EIN3. That is ethylene triple response 1, ethylene response factor 1, constitutive triple response 1, ethylene insensitive 2 as well as ethylene insensitive 3, that are the regulators of the signaling pathway. But what is the name of the receptor? It's going to be ETR1. So this receptor is acting as a ETR1 is acting both as a receptor as well as a regulator. Now what's the signaling pathway? It's bacterial two-component regulatory system. Now let us see what happens in the signaling pathway. So suppose this is a plasma membrane. Here the ethylene receptor, that is ETR1, is present in the form of dimer. So suppose this is ETR1 which is present in the form of dimer, but this is inactive dimer because there is no ligand for this receptor. And what's the name of the ligand? It's going to be ethylene. So they are inactive dimer that is associated with some copper ion because they need copper ion for their functioning. But in this receptor, there is a one domain that is what called as CTR1 domain. This is called as constitutive triple response 1 domain that is present in the cytoplasmic domain of this receptor. So when the signaling is off, in the absence of ethylene, such as ETR receptor will activate CTR1. That means if no ethylene is there, this CTR1 domain will be activated, which is a serine threonine kinase. And now if this is activated, this is a kinase domain. Means it will add phosphate group to its substrate. Now this CTR1 will add phosphate group to the substrate that is called as EIN2. This is a transmembrane protein that is what called as ethylene insensitive 2. So once CTR1 is activated, it will add phosphate domain to this. So now phosphate group is added to EIN2. Now this is activated. it will add utilizing one molecule of ATP that is converted to ADP in the process. So now phosphorylation of EIN2 will inhibit the proteolytic cleavage. Now there are some kind of proteases enzyme that cleaves EIN2 when the ethylene hormone is present. But now this is phosphorylated because the signaling is not on. This was phosphorylated by CTR1. Once this is phosphorylated, the protease enzyme will not act from outside, because this is now inhibited. They are phosphorylated. This EIN2 is phosphorylated, so it will not be acted upon by any kind of protease. There means no proteolytic cleavage of EIN2 will be seen. That means no C-terminal domain will be formed. As no cleavage is there, that will not migrate into the nucleus. So no cleavage of EIN2 will be there, so no peptide generation, so it will not migrate into the nucleus. So there is one transcription factor, that is EIN3 inside the nucleus, that is activated when the proteolytic cleavage peptide binds to this EIN3. So if EIN2 is not cleaved, so it will not bind to EIN3 transcription factor and the signaling will be off. So there will be no ethylene responses inside the cell. But what happens if the ethylene hormone is present? Again, in the presence of ethylene, this ETR1 signaling is repressed. Why this signaling is repressed? Because this CTR domain is now inactive, because it was active in the absence of ethylene. Now ethylene is present, the CTR domain is inactive because the receptors are repressed. They will not activate its cytoplasmic domain, that is CTR1, which is called as constitutive triple response. So now CTR1 is inactive. Once it is inactive, it will not be able to phosphorylate its substrate, or you can say a transmembrane protein, that is EIN2. So the non-phosphorylated C-terminus of EIN2 is cleaved. Now if this is not phosphorylated, suppose, if this is not phosphorylated, means proteases enzymes from outside, they are able to cleave this EIN2? Yes. So in the presence of ethylene, this EIN2 is cleaved into smaller fragment, and the C-terminal of this EIN2 is only responsible for the further step of signaling process. Now what happens? The non-phosphorylated C-terminus of EIN2 is cleaved by a protease. Yes, they are cleaved and moves into the nucleus. So the C-terminal part of this EIN2 will move inside the nucleus. So suppose this is nucleus, and here is a one transcription factor, that is EIN3, that is ethylene insensitive 3. To this, a C-terminal of EIN2 binds and will activate the transcription process. Now this peptide is generated because CTR1 was inactive. So protease has cleaved this EIN2 and the C-terminal fragment will bind to this EIN3. Now in the nucleus, the C-terminal of EIN2 inhibit EIN3 ubiquitinylation. Now EIN3 was ubiquitinated when there was no, you can say ethylene. Because there was no ethylene and C-terminal also was not binding. So it was going for 26S proteasomal degradation. Thereby no transcription was seen. But here EIN3 is binding to C-terminal peptide. Now there will be no 26S proteasomal degradation of this factor, and it will retain or persist inside the nucleus. That will bind to the DNA or you can say that will bind to the DNA segments or to the ethylene response element. So this will now bind to the ethylene response element, which will initiate the transcription of one factor which is known as ethylene response factor 1. And this transcription factor is only responsible for the generation of, or you can say for the induction of, ethylene induced responses in the plant. And with this, the functions that we have seen is all seen in case of plants. So all these functions are seen in the plant body once the transcription process is induced. So in order to start the signaling process, the ethylene must bind to the receptor. That will repress the signaling process and will inactivate its kinase domain, that is CTR1. And once CTR1 is inactivated, it will not be able to cleave its target, that is EIN2. And now this EIN2 will be cleaved by some other protease, that is from outside. Now the C-terminal part will be produced, This C-terminal will bind to the EIN3 transcription factor inside the nucleus and will activate this transcription factor, which will further bind to the response element in the DNA and will initiate the transcription of ethylene response factor, which is going to be the synthetic factor, or you can say precursor for the synthesis of ethylene. And with this, the ethylene induced responses are seen in the plant. So this is all about the ethylene biosynthesis, where we have seen the discovery, its a detailed biosynthetic pathway, what are the inhibitors and the stimulants involved, what is its functional profile, as well as the signaling mechanism, what happens in case of when the signaling is off, or when the ethylene is not present, or when the ethylene is present. So this is all about the topic. So thank you everyone for watching this session. If you like the session, do not forget to like, share and subscribe to the channel, that is Biotecnika. Meet you in the next video. Till then, bye-bye. Take care. Have a nice day. Happy learning.